At 8 a.m., you have had a very heavy breakfast before heading on to your lecture class. At 12 noon, you begin to feel a little hungry because you have not had any snacks at all in between. But you have still some amount of energy left in you to do your activities at least before the lunch break, where you can have your lunch at 1 p.m. Now, this happens to most of us quite frequently, but have you ever wondered how and what really pushes you through that moment? Now, we owe a lot to glycogen, which is the topic of this session. So, this session, I'll be talking about glycogen metabolism, and I'll also be talking how glycogen is utilized and how it is formed in our body. So, what is glycogen? Glycogen is a carbohydrate. It is a homopolysaccharide, meaning it contains of several sugar units, more than 10 sugar units, repeating units, and the sugar unit that is present over here in case of a homopolysaccharide, that is glycogen, is glucose. It is a storage form of glucose in animals, whereas we have starch also, which is a storage form of glucose, but it's present only in plants. It is also considered to be a non-reducing sugar, meaning it does not contain any free aldehyde or keto group that can interact with other compounds. Now, glycogen has two components. There's a straight chain and you have a branch chain. The same structure is also seen in starch. So the straight chain, normally you have glucose units like this, and each individual glucose units are linked together by an alpha-1,4 glycosidic linkage. Now, what do you mean by alpha-1,4 glycosidic linkage? It's nothing but the bond is formed between the first carbon atom of an alpha-D glucose and the fourth carbon atom of another alpha-D glucose. So this is how the bond is formed in case of alpha-1,4 glycosidic linkage. This straight chain is called as an amylose component. You can also have branch points at any level like this. The branch component is known as an amylopectin, and the bond formed at the branch point is an alpha-1,6 glycosidic linkage, meaning the bond is formed between the first carbon atom of an alpha-D glucose and the sixth carbon atom of another alpha-D glucose. So starch and glycogen has both these components. You have a straight chain and a branch chain, but the only difference between the two structures, that is in case of starch and glycogen, is that glycogen has more branched points in the structure. So this branching, the excessive branching which occurs within the glycogen, makes the entire molecule very compact, so that a lot more glycogen can be stored in our human body. The main site of storage for glycogen is the liver and the muscle, and the function of glycogen depends on the site at which it is stored. So the glycogen which is stored in liver is mainly used for energy purpose, especially during meals, especially when we don't consume meals between a particular time, like say between your breakfast and lunch. Our blood glucose level is maintained by the breakdown of glycogen which is stored in the liver. Whereas the glycogen that is stored in the muscle is only used for the purpose of deriving energy for muscle contraction. So now let's have a look how much of glycogen is stored in the liver and the skeletal muscle. So it is said that in a 100-gram tissue of the liver, around 10 gram of glycogen exists. Whereas in case of muscle, around 1 to 2 gram of glycogen is present in 100 gram of the tissue. But you know that the human body is made of a lot of muscle. So when you consider the entire muscle present in the body, it is said that at least 3/4 of the total glycogen content is stored in the muscle, and only a small amount is present in the liver. Now coming to metabolism, glycogen metabolism has two main pathways. One is glycogenesis. Genesis means to form, so there is formation of glycogen in glycogenesis. And then you have glycogenolysis. Lysis means to break. So breakdown of glycogen is known as glycogenolysis. Let's have a look at the first pathway, which is formation of glycogen or glycogenesis. So glycogenesis is defined as the formation of glycogen from glucose. Glucose is the monosaccharide which is being repeated to form glycogen. And the site where glycogenesis occurs is in the liver and muscle. This is the two places where glycogen is stored. The subcellular site is the cytoplasm. All the enzymes required for glycogenesis is present within the cytoplasm of the cell. The starting material obviously is glucose and the end material is glycogen. The main key enzyme whose activity influences the entire rate at which glycogenesis proceeds is glycogen synthase. So this is the main key enzyme. And what are the other requirements in addition to glucose and glycogen synthase? So you need another enzyme known as the branching enzyme which adds branches to the glycogen molecule. And then you need ATP for energy, and UTP is required, that is uridyl triphosphate is required for the formation of UDP glucose, which is the active form of glucose in this reaction of glycogenesis. And UDP glucose acts as a donor, donating glucose to the growing glycogen molecule. So let's have a look at the steps of the pathway. There are four main steps, starting with formation of UDP glucose. As I've told you earlier, UDP glucose acts as a donor of glucose for the glycogen. Next is formation of a glycogen primer. This is the foundation on which the entire glycogen molecule is built on. And then you have the main glycogen synthesis, and finally addition of the branch points. Now let's have a look at the steps one by one, starting with formation of UDP glucose. So for formation of UDP glucose, the starting material is glucose. So glucose undergoes phosphorylation in the presence of the enzyme either hexokinase or glucokinase. Hexokinase is present in the muscle, whereas glucokinase is present in the liver. Since this is a phosphorylation reaction, ATP is required for donating a phosphate group, and then it gets converted to ADP. The cofactor required for a phosphorylation reaction, that is in case of hexokinase and glucokinase, is magnesium. So now the phosphate from ATP is donated to glucose, and the phosphate gets attached to this 6-carbon atom of glucose, forming glucose 6-phosphate. Okay, the phosphate gets attached to this 6-carbon atom of glucose. Now in the next step, what happens is the phosphate which is attached to the 6-carbon atom gets shifted to the 1st carbon atom of glucose. And the enzyme that is required for this shifting is known as phosphoglucomutase. This is a reversible reaction. In the next step, we get glucose 1-phosphate. Now it becomes very easy from this step for the formation of UDP glucose. Now in the next step, which involves the formation of UDP glucose, I'll just write it down here. This is the last step, or the last substance formed in this step. So in order to form UDP glucose, we need a molecule of uridyl triphosphate, also known as UTP. So UTP is added to the reaction. UDP gets attached to glucose. Now, if you just imagine this UTP, it's just for you to understand. I can say that UTP gets split into UDP. There's 2 phosphates attached to uridyl, and then you have an additional phosphate group. So together, if you add UDP plus the phosphate group, you get UTP. So the UDP portion gets attached to the glucose, whereas you have one phosphate group over here and the other phosphate attached to glucose. Those 2 phosphates come out as a pyrophosphate. That means 2 phosphates joined together. And the pyrophosphate is then broken down by an enzyme called as pyrophosphatase. To give you 2 inorganic phosphate. The enzyme required for this conversion, that is glucose 1-phosphate to UDP glucose, is known as UDP glucose pyrophosphorylase. So this is the step, and this is how UDP glucose is formed, which acts as a donor of glucose for glycogen. Now we're done with activation of glucose. Now we'll move on to the next one, that is synthesis of glycogen primer. Now the primer over here is like a foundation. Now you have probably seen a building. For any building to stand erect, you need a foundation on which it is built on. So likewise, we need a glycogen primer which acts as a foundation, and on top of that you have the growing glycogen molecule which is built on. So let's have a look at synthesis of glycogen primer. Now let's see how this foundation or the glycogen primer is formed. First of all, you need a protein called as glycogenin. Now glycogenin is a protein. It's a dimeric protein, meaning it contains two subunits. I'll just draw the structure. Imagine this is glycogenin, and then there's a hydroxy group present on the protein. The hydroxy group is provided by the amino acid tyrosine, which is present in the structure of the protein. So this hydroxy group, which is present on the glycogenin, it serves as a point of attachment for glucose units. So a short stretch of glucose unit is formed, which constitutes the primer. How does that happen? Let's have a look. So you have glycogenin over here, and this is the hydroxy group, which serves as an attachment. Then you have UDP glucose, which is a donor of glucose. UDP glucose donates a glucose molecule to glycogenin, thereby releasing UDP. And so what happens is, this is your glycogenin. This is the hydroxy terminal. To the hydroxy terminal, you will have a glucose unit attached. So I'll just represent UDP glucose as something like this. Imagine this is UDP and glucose is a circle. Okay, so glucose is a circle, so I'm just going to add one circle over here. This is nothing but glucose. Now what happens is, this step will occur 7 times, so that a short stretch of glucose unit is formed. So this is the first glucose added. Then again UDP glucose will add another glucose. This is second one. Then you have a third glucose unit added, fourth one, fifth one, sixth, and then seventh. So like this you get a short stretch of glucose unit. This is known as glycogen primer. So we are done with the foundation. Now all we have to do is just add a lot more glucose molecules to form the glycogen molecule. The main step, glycogen synthesis. So I'll just draw the glycogen primer once again. We have the glycogenin protein. To that we have an oxygen group and then 7 molecules of glucose added. And remember, this is 1, 2, 3, 4, 5, 6 and then 7. The bond formed between the individual glucose unit is an alpha 1,4 glycosidic linkage. That is one thing you need to remember. Okay. Now once the glycogen primer is there, what happens in the next step is a straight chain or an amylose component of the glycogen is formed. This occurs in the presence of the enzyme glycogen synthase, which is the main key regulatory enzyme involved in glycogenesis. So we have glycogen synthase acting at this point. This is the main key enzyme. And then you have UDP glucose, which is the donor of glucose. It keeps on adding to the primer glucose units. And at the same time, what happens is UDP is released. So this cycle occurs several times till a long chain of amylose component is formed. So I'll just draw this once again here. We have the primer first. And then we have the new glucose units being added here sequentially by glycogen synthase one by one with the linkage of alpha 1,4 glycosidic bonds formed between the successive glucose unit. So this keeps on going, and the new glucose units I've just colored it, darkened it, so you can have a look at it and you can notice a difference. These are the new glucose units and this is part of your glycogen primer. Now, what really happens here is the glycogen synthase keeps on acting, keeps on adding a new glucose unit by forming an alpha 1,4 linkage with the terminal glucose, the last glucose present in the chain. And then what happens is the chain length is increased till about 11 to 12 glucose units are present in the straight chain. Then what happens is once that straight chain is lengthened enough till around 11 to 12 glucose units are present, a part of the straight chain is then shifted to a branch point, forming a branch. So that's the next step. So till here, remember this is the straight chain that is formed. And the individual bonds formed between the glucose unit is an alpha 4 linkage. Now let's have a look at how the branch point is formed. So that's the next step, formation of branches. So in the earlier step you have seen there was a straight chain like this. Now the next enzyme comes into play. The next enzyme which is involved in adding the branch is known as a branching enzyme. So what does this branching enzyme do? The branching enzyme simply takes units around 6 to 8 glucose unit from one end and shifts it to a branch point. Let's see. Now say for example 6 units are taken from here. 1, 2, 3, 4, 5, 6. So till here there will be a break, there will be a cut in the structure, linear structure, so that portion will be shifted to a branch point. So you'll get something like this. This is glycogenin, and then you have the primer. And then you have 3 of those new glucose units which was added by glycogen synthase. And then the remaining 6 glucose units are shifted to a branch point somewhere here. So the bond that is formed at the branch point by the branching enzyme is known as alpha 1,6 linkage. This is how a branch point is added. Now what happens in the next step is you have both the glycogen synthase and branching enzyme acting one by one, so that a straight chain is formed, then a branch point is formed, straight chain, then branch point, and this keeps on going on till a huge glycogen molecule is formed. So you have repeated cycles in which both glycogen synthase and branching enzyme act one at a time. And then you will have the end product, which is glycogen, after several steps have occurred like this, where you have straight chain, branch branch points happening, and then you will get something like this. So you will have glycogenin, the protein, at the center or the core, and then you will have a structure like this. With several branch points. More number of branch points makes the structure even more globular and more compact so that it can be stored within the liver or muscle. This structure, compact or globular structure, is known as glycogen, which is the storage form of glucose. So this is how glycogenesis occurs. Now we'll move on to the next one, that is glycogenolysis. In other words, breakdown of glycogen to form glucose. Now the main site where glycogenolysis occurs is where glycogen is stored. So you have liver and muscle. And again, the subcellular site is cytoplasm. Starting material obviously is glycogen, but the end products will vary depending on where the glycogen is being broken down. So in case the glycogen is being broken down in the liver, the end product will always be glucose. But if the breakdown is occurring within the muscle, then the end product will be glucose 6-phosphate. Why the difference? Now, main difference is because muscle lacks a particular enzyme which is involved in the breakdown, or which is involved in conversion of glucose 6-phosphate to glucose. So what is that enzyme? I'll come to that later when I'll be talking about the reactions. And the main regulatory enzyme of the pathway is glycogen phosphorylase. The main key enzyme in glycogenesis was glycogen synthase. Here it is glycogen phosphorylase. So what are the steps? Yes, before going on to the steps, these are the other enzymes required. So in addition to glycogen phosphorylase, the key enzyme, you need a debranching enzyme to remove the branches from the glycogen. You need a phosphoglucomutase to, convert glucose 1-phosphate to glucose 6-phosphate. And then you need a glucose 6-phosphatase to convert, or to obtain rather, glucose in the end. So let's have a look at the steps first. So the first step is the action of glycogen phosphorylase, where glucose units are removed. And then you have a debranching enzyme which removes the branches. And then finally formation of glucose 6-phosphate or glucose, depending on the site where the reactions occur. So let's have a look at action of glycogen phosphorylase. So now just draw a structure, simple structure of glycogen, not the globular one that I drew earlier. Imagine this is the straight chain. And then you have a branch point somewhere here. So I'll just to demarcate the branch point, I'm going to draw this and then some blue circles here. This is nothing but glucose units. Okay. Now the first step is glycogen phosphorylase enzyme acts on this. And what does glycogen phosphorylase do? Glycogen phosphorylase removes the additional glucose sequentially from either ends of the branching point, so that there are only 4 glucose units on all sides of the branching point. So leaving behind 4 glucose units on either side of the branching point, rest of the other glucose molecules are released by glycogen phosphorylase in the form of glucose 1-phosphate. So let's see what are the additional glucose molecules that you can see here. So you can see, as I told you, 4 glucose units have to be there. So this portion should be there, attached to the branch point. This portion should be there, and then this portion should be there. So the additional ones are, additional glucose molecules are the one here, over here, okay, and then the one on top. So all those glucose molecules, the one here, I'll just write here, 1. Then you have 1, 2, 3, 4. You have 2 extra glucose molecules there. And then you have 1 glucose molecule on top, which is an extra. All these glucose molecules are released in the form of glucose 1-phosphate. Now what are we left with? Let's have a look at the structure. So we are left with something like this. Now if you have a look, if this is the branch point, you have 4 units of glucose to your left, 4 glucose units to your right, and 4 glucose units to the top. So this is the end thing that should happen after glycogen phosphorylase acts. There should be 4 glucose units on any side of the branch point. Now, once this is done, we have the next enzyme acting, which is debranching enzyme, where the branch point is removed. So let's have a look at how that happens. So in order to understand how the branch points are removed, we need to be aware that debranching enzyme has 2 activities. It's known as dual functions of debranching enzyme. So debranching enzyme is said to have a glucan transferase activity, which simply means that the enzyme is concerned with the transfer of a glucose group, and the other activity which is shown by debranching enzyme is a alpha 1,6 glucosidase activity. Glucosidase means removal of a glucose, and alpha 1,6 means removal of the glucose at the branch point where the alpha 1,6 linkage exists. So now let's look at the action of debranching enzyme. For this, I will draw the structure that was formed at the previous step, which is something like this. And then you had the branch point here. Yes. Now this, I forgot to mention in the previous slide, this is a partially digested or partially broken down product of glycogen. So it is called as limit dextrin. Limit dextrin is nothing but a branch oligosaccharide. Now in the next part, we have the debranching enzyme acting here. And the first enzyme or the first function of the debranching enzyme to occur is the glucan transferase. So what does the glucan transferase do? Glucan transferase mainly transfers a trisaccharide from the branch point to the straight chain. What is the trisaccharide over here? Trisaccharide is the 3-glucose unit. So this 3-glucose unit that we see here, 1, 2 and 3, these 3-glucose unit, also known as a trisaccharide, is shifted from the branch point to the straight chain. So you will get a structure something like this. And then you have the 3 sugar units being added to the straight chain. And you're left behind with 1 glucose unit at the branch point, which is this. So this is glucan transferase acting here. Now the debranching acts on this structure once again, and the next enzyme or the next activity of the debranching enzyme functions, which is alpha 1,6 glucosidase. So what does this alpha glucosidase do? Alpha glucosidase or alpha 1,6 glucosidase removes the glucose, the last glucose, which is attached to the linear chain, and it forms a bond that is alpha 1,6 linkage. So that alpha 1,6 linkage is broken down so as to release the glucose unit present at the branch point. So this glucose unit gets released, and it is released as glucose. Mind you, it is not glucose 1-phosphate but free glucose, which is formed using debranching enzyme. In case of glycogen phosphorylase, the glucose units are always released as glucose 1-phosphate. So what is left once this glucose is removed? You're left with just the linear chain. Now it becomes very easy for glycogen phosphorylase to act on this. And glycogen phosphorylase removes each glucose unit as glucose 1-phosphate. So if you have, say for example, there is around 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. There are 12 sugar units in the linear chain. Glycogen phosphorylase will act 12 times to remove the glucose unit one by one sequentially, so that you have glucose 1-phosphate released till you are left with none. Now what happens to this glucose 1-phosphate? So that is explained by the last step, that is formation of glucose 6-phosphate and glucose. So we are left with a lot of glucose 1-phosphate that is formed with the help of glycogen phosphorylase. So I'll just write this down here. Glucose 1-phosphate. Glucose 1-phosphate undergoes an isomerization step where the phosphate from the first position of glucose is shifted to the 6th position. This is a reversible reaction. catalyzed by phosphoglucomutase. So the phosphate which was attached to the first carbon atom is now just simply shifted to the 6th carbon atom, leading to the formation of glucose 6-phosphate. Now the formation of glucose 6-phosphate is a very crucial point, and it also plays a major role in two things which I'll be talking about now. So now, depending on where this glycogenolysis is occurring, whether it's occurring within the liver or the muscle, you have two different set of products formed. So in case this is occurring within, glycogenolysis is occurring within the liver has an enzyme known as glucose 6-phosphatase, which is also a key enzyme of gluconeogenesis, in which the phosphate group which is present or attached to the 6th carbon atom of glucose is removed. Water molecule is required for the removal of the phosphate group, and the phosphate group is removed. So this will leave us with just glucose, and glucose enters into the bloodstream and then it is utilized by various cells. So this is how glycogen is broken down in the liver and it is utilized for formation of glucose, which enters in the bloodstream, taken up by various cells and utilized for energy by those cells. The other thing is, if supposing glycogenolysis occurs within the muscle, what happens in the muscle is, this enzyme glucose 6-phosphatase is absent in the muscle. The muscle lacks glucose 6-phosphatase. So the muscle is unable to break down glucose 6-phosphate. So what happens is, the glucose 6-phosphate directly enters into a pathway known as glycolysis till it forms pyruvate. But you know, in muscle, it is anaerobic glycolysis which takes place. So pyruvate is further converted to lactate. And through the process of anaerobic glycolysis within the muscle, ATP is generated, which is utilized for muscle contraction. The energy is utilized for muscle contraction. Now, another thing is, this glucose 6-phosphate, now this is about glycogenolysis that I've spoken about in the liver and muscle. In addition to all this, glucose 6-phosphate can also be taken up by other cells, where it can enter into a pathway known as HMP shunt, hexose monophosphate shunt, wherein it can form certain pentose sugars, that is ribose or ribose 5-phosphate, and an energy reducing equivalent that is known as NADPH. These are the two important products of HMP shunt. You have a ribose sugar which is formed and you have NADPH which is formed, which is a reducing equivalent. Now ribose 5-phosphate can be utilized for the formation of nucleic acids, that is DNA and RNA, which make up the genetic constituent of or genetic framework of an individual. So this is an important thing that you need to remember, because there is a disorder that is one of the examples of glycogen storage disorder, where I'll be talking about this aspect of glucose 6-phosphate entering into the HMP shunt. So that was it about glycogenolysis in this liver and muscle. Now, a small amount of glycogen is also broken down within the lysosomes. Lysosome is a cellular organelle, and the lysosomes are also called as suicide bags, because it contains certain hydrolytic enzymes that are involved in breaking down of certain substances. So there is a hydrolytic enzyme or a hydrolase present in the lysosome, which helps break down glycogen within the lysosome. The enzyme is known as acid maltase or alpha-1,4-glucosidase. Now, the significance of this pathway is not really clear. That's why it's not spoken about much. But yes, a small amount of glycogen is still degraded within the lysosome. And deficiency of that enzyme, of that lysosomal enzyme, leads to a disorder called as Pompe's disease, which is an example of a glycogen storage disorder, which I'll be talking about in a little while later. Now, coming to significance of glycogen metabolism as such, Now, if you understand this basic concept, it will be much clear to you how well the glycogen metabolism is regulated. So, in the well-fed state, when we take in any food, obviously, after a period of time, there is going to be an increase in the blood glucose level following intake of food. What should happen to this glucose? Glucose that is present in blood. Yes, some amount of glucose will be taken up by the cells, and some amount of glucose will be stored within the liver or the muscle as glycogen. So you have a process that is glycogenesis that will take place in the well-fed state for the formation of glycogen in the liver or muscle. This occurs in well-fed state. Now, what happens in the fasting state? Yes, you have not eaten any food for quite some time. Your blood glucose will tend to decrease after a period of time. So this is when glycogen comes to our rescue. Glycogen, especially that is stored in the liver, comes to our rescue and helps increase the blood glucose level by the process of glycogenolysis. So remember, glycogenesis always occurs within the well-fed state when you have eaten some food, and glycogenolysis will always occur, especially in the fasting state or when we are involved in some kind of an exercise where there is active muscle contraction. So these are the two states that you need to remember. So overall, this is what is the significance of glycogen metabolism. Coming to regulation, now glycogenesis and glycogenolysis, these two pathways will never occur at the same time. When one pathway operates, the other will not. This is done in order to prevent any wastage from happening or from letting any futile cycles from happening. Futile meaning waste. So a lot of wastage is overcome just by regulating these two pathways at 3 levels. So at the level of covalent modification, allosteric regulation, and influence of calcium, especially on muscle glycogenolysis. These are the three levels at which both glycogenesis and glycogenolysis are well regulated. And the two main key enzymes that are involved in this regulation is glycogen synthase from glycogenesis, and you have glycogen phosphorylase, which is the key enzyme of glycogenolysis. So now let's have a look at the first aspect, that is hormonal regulation or covalent modification. So under hormonal regulation, we have hyperglycemic hormones and hypoglycemic hormones which regulate the activity of those two enzymes, glycogen synthase and glycogen phosphorylase. So like I've told you earlier, during the well-fed state, you should have glycogenesis happening, and during the fasting state, you should have glycogenolysis happening. So during the well-fed state, you have blood glucose increasing. Obviously, the pancreas will be stimulated to release a hormone known as insulin. Insulin will allow the uptake of glucose by various cells, and at the same time, it will also stimulate the process of glycogenesis. Remember, insulin is a hypoglycemic hormone. So this is what happens during the well-fed state. Now, in a fasting state, the blood glucose level will be decreased or low. So you have certain hyperglycemic hormones that get secreted, like glucagon or even epinephrine, especially when we start running out of fright. Epinephrine is supposed to be a hormone that is secreted especially at fear or fright. So those two hormones, hyperglycemic hormones, I'll just write glucagon over here, stimulate glycogenolysis. So that glycogen is broken down and then glucose is formed, and that can help maintain the blood glucose level. In case of epinephrine, which is secreted in fear or fright, you start running, especially when you see a dog chasing you or something like that. That time you have epinephrine which is released. Epinephrine will stimulate glycogenolysis in the muscle, and because of muscle contraction, ATP is generated, glycogen is broken down, giving energy in the form of the glucose which is being oxidized to lactate, and that's how you're able to run. So this is a brief overview of how the hormones act. Now, if you understand this key concept, it is easy to understand how covalent modification occurs. Now, this is what I've just written here, and I've explained in the form of a flow chart. Now, there are two key enzymes, but before I go into that, you need to know that in covalent modification, if you need to write that an enzyme is in the active form, you write the name of the enzyme, like say for example, glycogen synthase is the name of the enzyme, followed by hyphen, and the active form is always represented by the symbol A. So this is called active form of the enzyme. If you have to show that, you show an inactive form of glycogen synthase, you just have to write the name of the enzyme, glycogen synthase, followed by hyphen, and then B. B means it's an inactive form. Likewise, you can do the same for any of the enzymes to represent it as active or inactive. Just remember to add the A for active. Now, let's see what is the active form of both the enzymes, glycogen synthase and glycogen phosphorylase. The active form of glycogen synthase is said to be the dephosphorylated form. That means phosphate is not attached to the structure of the enzyme. This is the active form of glycogen synthase. Whereas in case of glycogen phosphorylase, the active form is the phosphorylated form. That means phosphate is attached to the structure of the enzyme. So a simple way to remember is phosphorylase, glycogen phosphorylase is always active in the phosphorylated form. This is one way you can remember which one is active, dephosphorylated or phosphorylated. So phosphorylase is always active in the phosphorylated form, whereas the reverse is true for glycogen synthase, where synthase exists as an active form in the dephosphorylated form. Now let's have a look at how covalent modification occurs. We'll start off with glycogen synthase. So initially glycogen synthase is in the inactive form. So I'll just write B over here, and then I want to convert this to an active form, which is glycogen synthase A. Remember, the active form of glycogen synthase A is the dephosphorylated form. So I'll just write DE capital P, meaning phosphate is not attached. Whereas glycogen synthase B, the opposite is true. The inactive form is the phosphorylated form. Now, how do you convert phosphorylated to dephosphorylated? Simply, you have to remove the phosphate group from the enzyme. And to remove the phosphate group, you need a phosphatase enzyme. The phosphatase enzyme uses a water molecule and removes the phosphate from the enzyme. Okay, we need the enzyme phosphatase here. Okay, and in order to convert dephosphorylated to phosphorylate, you need to add a phosphate group. And who usually does that? It's a kinase who adds a phosphate group. So I'll call this as glycogen synthase kinase, and ATP acts as a donor, adds a phosphate group, and you have ADP here. Now, glycogen synthase should occur within the well-fed state where excess glucose is stored as glycogen. And in the well-fed state, you know that insulin level has to be secreted. So I can say that insulin stimulates the activity of phosphatase, thereby causing the dephosphorylated or active form of glycogen synthase to be formed. Whereas glucagon or even epinephrine. These are the two hyperglycemic hormones. These two will stimulate glycogen synthase kinase, causing phosphorylation to happen, as a result of which this becomes the inactive form and glycogenesis gets inhibited. So insulin stimulates glycogenesis by increasing the activity of glycogen synthase. On the other hand, glucagon and epinephrine activates the phosphorylated form, which is the inactive form, and inhibits glycogenesis. So this is what really happens during fasting and well-fed state. This is in the well-fed state and this is the fasting. So this was about glycogenesis. Now let's have a look at glycogenolysis. Again, we'll first just write down the name of the enzymes. Glycogen phosphorylase, and this is the inactive form. The inactive form is in dephosphorylated state. And then I'll just write here glycogen phosphorylase A, which is the active form. The active form is in the phosphorylated form. Phosphorylase for phosphorylated form. Okay. Now, in order to convert this again, you need a phosphatase. Sorry. In order to convert this, you need a kinase. Because you have a dephosphorylated form, you need to add a phosphate group. So you need a kinase for this. Kinase utilizes ATP, which acts as a donor for phosphate. The phosphate is attached to glycogen phosphorylase, and then you get a phosphorylated form which is active. Kinases are always stimulated by glucagon or epinephrine. So in the presence of glucagon or epinephrine, kinases are activated. You have the phosphorylated form of glycogen phosphorylase which is formed, which is the active form. So glycogenolysis occurs in the presence of glucagon or epinephrine, especially in the fasting state. So that glycogen is broken down and then glucose is formed, which is utilized for energy. Likewise, to convert phosphorylated form to dephosphorylated form, you need the enzyme phosphatase, which removes the phosphate group. So water molecule is required to remove the phosphate group, and the enzyme is known as phosphatase. Phosphatase is always stimulated by insulin, and insulin is secreted during the well-fed state. So in the well-fed state, phosphatase is stimulated, the dephosphorylated form is activated, and you know dephosphorylated form is the inactive form. Therefore, you will not have glycogenolysis happening when the person is said to be in well-fed state. So this is a brief overview of how covalent modification occurs, and it is under the influence of the hormones, insulin and glucagon. So just a brief summary. I've just put both the enzymes here, and from this you can come to know about the reciprocal regulation of both the enzymes. So you have glycogen synthase. I'll just put both the hormones here. You have insulin here and you have glucagon here. Likewise, I'll just write insulin and glucagon there. And we know that the active form of glycogen synthase is the dephosphorylated form, and the active form of glycogen phosphorylase is the phosphorylated form. So I've put P. Okay. So in the presence of insulin, phosphatase is stimulated. Phosphatase removes a phosphate group. Thereby, it stimulates glycogen synthase so that glycogenesis can occur. At the same time, glucagon stimulates kinases. Kinases are known to add a phosphate group. So, phosphorylated form, if glycogen synthase is seen in phosphorylated form, it is said that it is inhibited. So, in the presence of glucagon, the enzyme glycogen synthase is inhibited. Likewise, on the other hand, insulin stimulates phosphatase. Phosphatase removes the phosphate group. So, minus P I'll write here. So, that should not happen because a dephosphorylated form of glycogen phosphorylase is an inactive form. So, I'll say that in the presence of insulin, glycogen phosphorylase is inactive. And then glucagon stimulates kinases. Kinase adds a phosphate group. Phosphorylated form of the glycogen phosphorylase is active. Hence, in the presence of glucagon, glycogen phosphorylase becomes active. So, this is the reciprocal regulation of glycogen formation and glycogen breakdown. The next one is allosteric regulation. So, according to that, the availability of substrates to form glycogen, it could be either glucose or glucose 6-phosphate. These are two substrates utilized, and the presence of energy. If you have those high energy and presence of substrate, glycogenesis gets stimulated, that is formation of glycogen, whereas the reverse happens with glycogenolysis. Let's have a look at the diagram. So this again is the same thing. We have glycogen synthase and glycogen phosphorylase, the two key enzymes. So like I've said, the availability of substrates and high energy compounds are required for stimulating glycogenesis or the enzyme glycogen synthase. So the substrates could be glucose or glucose 6-phosphate. These two can stimulate the activity of glycogen synthase, thereby promoting glycogenesis to occur. And the presence of high energy compounds in the form of ATP will also trigger the glycogen synthase to act so that glycogenesis occurs. So these are the key factors that promote glycogen synthase activity. The reverse is true for glycogen phosphorylase. The substrates, glucose as well as glucose 6-phosphate, these two will inhibit glycogen phosphorylase if it is present, and high energy compounds will inhibit glycogen phosphorylase. On the other hand, if you have a look here, presence of AMP, which indicates that it is low energy within the cell. AMP indicates low energy. So low energy will stimulate glycogen phosphorylase to occur, and the presence of calcium, especially in the muscles where glycogenolysis is occurring, calcium will activate the activity of glycogen phosphorylase so that glycogenolysis will occur, and calcium is usually secreted during muscle contraction. Now let's have a look at the effect of or the influence of calcium on glycogenolysis. What happens during muscle contraction is calcium is usually released from the sarcoplasmic reticulum, and when calcium is released from the sarcoplasmic reticulum, it binds to a protein known as calmodulin and forms a complex called as calcium calmodulin complex. This complex activates phosphorylase kinase, and phosphorylase kinase is known to add a phosphate group to the glycogen phosphorylase enzyme, and the phosphorylated form of glycogen phosphorylase is the activated form. So it activates glycogen phosphorylase, thereby promoting glycogenolysis. That's how calcium acts on muscle glycogen. So the influence of calcium is strictly meant for glycogenolysis in the muscle. Now moving on to the disorders concerning glycogen metabolism. The disorders are known as glycogen storage disorders, and more or less it is about the accumulation of abnormal glycogen or abnormal quantity of glycogen within the different tissues. So there are 7 major types of glycogen storage disorders, though there are plenty more other disorders. I've not listed them out here, but these are the major ones. So it starts with type 0. Now type 0 is not per se a glycogen storage disorder. There is no accumulation of glycogen in that. Rather, there is a deficiency of glycogen formed. So this is one exception. That is why you don't have a number here. Rather, it is called as type 0 because there's deficiency of glycogen and not accumulation of glycogen or any of its products. Now, type 1 is Von-Gurk's disease. Type 2 is Pompe's disease. Type 3 is called as Cory's disease or limit dextrinosis because of the accumulation of limit dextrins, or it's also called as Forbes disease. Type 4 is called as Anderson's disease or amylopectinosis because of the accumulation of amylopectin. And type 5 is called as McArdle's disease, and type 6 is called as Her's disease. Now, there's a very easy way to remember the disorders with the exception of type 0. I think even if you don't include type 0 under glycogen storage disorders, it's fine. But if you remember the next one, that is from type 1 to type 6, it's more than enough. So there's one mnemonic, that is versatile player Cory Anderson makes history. Now, who is this Cory Anderson? Cory Anderson is a New Zealand cricket player. He made history in 2014 by scoring the fastest century in a one-day international cricket match. Now, I've just added his name here, and Cory obviously will be Cory's disease and Anderson will obviously be Anderson's disease. So you're left with only V, P, M and H. So V stands for Von Gurk's disease. P stands for type 2, which is Pompe's disease. Cory is Cory's disease, but make sure you do not do the mistake. It is C-O-R-I disease. Anderson is Anderson's disease. And here the son is not S-O-N but S-E-N. And then M stands for McArdle's disease. And then finally you have H, which stands for Her's disease. So this is how you can remember the entire glycogen storage disorders by remembering this mnemonic. Let's have a look at each of the disease individually, starting with type 0, which is the deficiency of glycogen synthase. Glycogen synthase is a key enzyme in glycogen formation. So if the enzyme is deficient, what is going to happen? You're not going to have any glycogen stored in the liver or the muscle. So here mainly the defect is with the glycogen synthase in the liver. Hence, there will be deficiency of glycogen in the liver. So what happens is after you take a meal, after you eat some food, there will be a sharp increase in blood glucose level. And that is called as transient hyperglycemia, a sharp increase in blood glucose level. And obviously, in a normal person, what should happen is insulin should come into action and glucose should be utilized for glycogen formation and stored in the liver. That happens under normal condition. But here, because of the deficiency of synthase, glucose is not channelized towards glycogen formation. And what happens is in the presence of insulin will tell the glucose, okay, you have to be taken up by the different cells for energy purpose. That will be done. Glucose will be channelized into different cells, taken up by different cells, utilized for energy. But the other aspect, that is glucose being channelized towards glycogen formation, does not occur. So glycogen is not formed, as a result of which, especially between meals, when the blood glucose tends to decrease and when glycogenolysis should occur, there's no glycogen available for the glycogen to be broken down to give you glucose and maintain the blood glucose level. So between meals especially, there will be hypoglycemia because of non-availability of glycogen. The next one is type 1 disease. This particular disorder can be split into 1A and 1B. So 1A is called as Von Gurk's disease, and the deficiency here is glucose 6-phosphatase. Glucose 6-phosphatase in the liver is concerned with the conversion of glucose 6-phosphate to glucose, so that glucose is utilized, sent into the bloodstream and utilized for energy purpose by different cells. So because of that, the conversion doesn't occur. Glucose 6-phosphate does not get converted to glucose. There's fasting hypoglycemia. Along with that, there is hepatomegaly because of the accumulation of glucose 6-phosphate within the liver. There is lactic acidosis. Why? I'll come to that later. Hyperuricemia, increase in blood uric acid level. Why again? That will be explained a little while later. And then hyperlipidemia, increase in the serum lipid levels. So that was it about Von Gurk's disease, that is type 1A. 1B doesn't have a name, but it's supposed to be a type of von Gog's disease, a rare type, where there's a defect in the transporter in the endoplasmic reticulum for glucose 6-phosphate. So the symptoms are almost same like von Gog's, but you have two more additional features, that is neutropenia, decrease in the neutrophil count. And because of the decrease in neutrophil count, these individuals will be highly susceptible towards infections. So they'll have recurrent infections. Now, I would just like to show you a brief outline of what really happens in von Gog's disease. If you understand this mechanism, you will be able to recollect what the manifestations are and why do they happen. So in von Gog's disease, the basic deficiency or the defect is in the enzyme glucose 6-phosphatase. So glucose 6-phosphatase, let me write here, glycogen is broken down to give you glucose 1-phosphate, and then glucose 1-phosphate undergoes a mutation to give you glucose 6-phosphate. Now this point is very crucial. Glucose 6-phosphate is normally in the liver broken down by glucose 6-phosphatase. We all know that, and then glucose is formed. But in von Gog's disease, the enzyme glucose 6-phosphatase is deficient or defective. So you won't have this reaction occurring only in the first place. And since the reaction doesn't happen, glucose is not formed, hypoglycemia occurs. Now, when hypoglycemia occurs, the pancreas is stimulated to release a hormone, hyperglycemic hormone, that is glucagon. And glucagon has said to have a stimulatory effect on an enzyme called as hormone-sensitive lipase, especially in the adipocytes. The adipose tissue is a site where all the lipids are stored in the form of triacylglycerol, and hormone-sensitive lipase basically breaks down that fat which is stored in the adipose tissue and causes release of fatty acids and glycerol into the circulation. So because of the increased activity of glucagon, it stimulates hormone-sensitive lipase, that is lipolysis, fat breakdown occurring within the adipose tissue. The fatty acids are released into the bloodstream. Fats are released into the bloodstream, leading to hyperlipidemia. So this is one aspect. Then in the muscle, let's see what happens. Now, obviously, you know, in the muscle, there is no glucose 6-phosphatase, but deficiency of glucose 6-phosphatase will lead to an accumulation of glucose 6-phosphate. So the glucose 6-phosphate, what will happen is in the muscle itself, okay, some of them might get transported in the muscle. It undergoes anaerobic glycolysis. You have glucose 6-phosphate entering into glycolysis, forming pyruvate, and then finally forming lactate. So there is increased lactate production leading to a condition called as lactic acidosis. A typical manifestation seen in von Gog's disease. So this is one aspect. So we are done with 3 of the clinical features. Now the next thing I told you when I was just talking about what happens to glucose 6-phosphate after glycogenolysis has occurred, I told you that glucose 6-phosphate in some cells can enter into a pathway called as HMP shunt. So HMP shunt, the main products are a ribose sugar, which is usually ribose 5-phosphate, a pentose sugar, which is ribose 5-phosphate, and NADPH, which is a reducing equivalent. Now because of the accumulation of glucose 6-phosphate, you will have a high amount of ribose 5-phosphate formed, and there will be increased production of nucleic acids. And because of the increased production of nucleic acids like DNA and RNA, they ultimately have to be broken down. So once they get broken down, they form uric acid. But there will be larger than normal production of uric acid in von Gog's because of the accumulation of glucose 6-phosphate, which gets converted to ribose and then nucleic acids and then uric acid. So you have increased uric acid formation leading to hyperuricemia. So this is how you can explain all the clinical manifestation of Von-Gurk's disease, and hopefully you'll never have to by heart the symptoms or the clinical manifestations of the disease. Now we'll move on to the next one, that is Pompe's disease. I've already mentioned when I was talking about the breakdown of glycogen within the lysosome. So the defective enzyme is a lysosomal enzyme itself called as acid maltase or alpha-1,4-glucosidase. And because glycogen is not broken down within the lysosome, there is accumulation of glycogen inside the lysosome, especially in tissues such as the heart, liver and muscle, leading to different manifestation. So if there's accumulation within the liver, it will lead to liver failure. Within the heart, it will lead to heart failure. And if there's accumulation within the lysosomes present in the muscle, it will lead to myopathy or muscle weakness. The next one is Cory's disease. Now, Cory's disease is also called as limit dextrinosis because there is the deficiency is mainly by debranching enzyme, and debranching enzyme acts on limit dextrins. So because of the deficiency of this enzyme, there is accumulation of limit dextrins. They are nothing but highly branched oligosaccharide, and since there is accumulation of limit dextrins, especially in the liver, there will be hepatomegaly. The liver will get enlarged. The liver will lose its function. There will also be fasting hypoglycemia because the whole point of glycogenolysis has stopped at limit dextrin. The further reaction that involves breakdown of glycogen doesn't occur to give you glucose. So obviously fasting hypoglycemia occurs, and if this limit dextrin gets accumulated within the muscle during muscle glycogenolysis, it can lead to accumulation within the muscle causing myopathy or muscle weakness. And there's one easy way to remember the enzyme concerned with Cory's disease. Always remember CD. C for Cory and D for debranching enzyme. Okay, so this is how you remember the name of the enzyme related to Cory's disease. CD, C for Cory, D for debranching enzyme. The next one is Anderson's disease or amylopectinosis. It's called amylopectinosis because of the accumulation of amylopectin within the concerned tissue. So the enzyme defect is branching enzyme, and again, if amylopectin gets accumulated within the heart muscle, it leads to cardiac failure. If it gets accumulated within the liver, liver failure. But very quite often what happens is those who have Anderson's disease usually die by 5 years of age because of cardiac failure. And an easy way to remember the enzyme defect with the disorder is AB. So remember, A stands for Anderson and B stands for branching enzyme. So CD and AB. The next one is McArdle's disease, and the defective enzyme is muscle phosphorylase. Remember, M for McArdle, M for muscle. So this is how you can remember the enzyme defect with the name of the disorder. Since muscle phosphorylase is affected, phosphorylase is concerned with the breakdown of glycogen. And since this occurs within the muscle, there is accumulation of glycogen within the muscle. As a result of which, individuals with this disease have an intolerance towards exercise. They won't be able to do even the basic of simplest exercise. And then the last one, that is type 6, is called as Her's disease. H for Her and H for hepatic or liver. So the concerned enzyme is liver phosphorylase, H for Her's and H for hepatic or liver. Liver phosphorylase is a defective enzyme. Obviously, glycogen will accumulate within the liver because of the deficiency of phosphorylase in the liver, and that will lead to enlargement of the liver because of accumulation of glycogen and leading to liver failure. And obviously, glycogen is not broken down, so there won't be glucose formed and hypoglycemia will result. So this is the way you can remember glycogen storage disorder. So with that, we're done with this session. Briefly to summarize, I spoke about glycogenesis and glycogenolysis. Glycogenesis is the formation of glycogen from glucose, occurs within the liver and the muscle, especially in the cytoplasm. And the key enzyme is glycogen synthase. There are 4 main steps: is activation of glucose, there is formation of a primer, glycogen primer, then formation of glycogen, the straight chain, and then the branch points are added. And then in case of glycogenolysis, it occurs within the liver or the muscle. Cytoplasm is the subcellular site. It's a breakdown of glycogen to form glucose. And there are, the steps that are involved is, first of all, the action of glycogen phosphorylase. Then you have the debranching enzyme coming into action, and then formation of glucose 6-phosphate or glucose, depending on the site at which glycogenolysis occurs. Then talking about the regulation of glycogenesis and glycogenolysis, the key enzymes of both the pathways are known. It is glycogen synthase in case of glycogenesis and glycogen phosphorylase in case of glycogenolysis. And there, at three levels, both the pathways are regulated in order to prevent futile cycles. So the three main ways through which regulation occurs are covalent modification or hormonal regulation, allosteric regulation, and last but not the least is influence of calcium, especially on muscle glycogenolysis. And then finally, I spoke about glycogen storage disorders, which mainly is concerned due to accumulation of abnormal glycogen or abnormal quantity of glycogen within the different tissues. So you have, I've spoken about 7 major types. You have type 0, which is glycogen synthase deficiency, type 1, which is von Gerk's disease, type 2, Pompey's, type 3 was Cory's, type 4 was Anderson's, type 5 was McArdle's disease, and type 6 is Her's disease. So with that, we are done with this session.